Okay, then I will start welcoming everyone to this session today of Amplifying Voices. This is the last session before our summer break. We will have a 3-month summer break and we'll come back to you in, I think it should be with September. And till then, I hope you all have a good time. And I also hope that you enjoy our last session today. We have a last-minute change of schedule here. Our second speaker today is someone who we actually didn't announce, and it's a kind of surprise, but we'll come back to that after our first speaker. And I'm very happy to welcome Roudaina Boukheloua, who is from Algeria and has a master's degree in biology and physiology with a focus on genomics and environment and genomics and biodiversity. And since 2020 is a PhD at the Institute of Hydrobiology at the University of South Bohemia in Czech Republic. And she is working with some organisms that she apparently is pretty amazed by. And I guess we also will be after her presentation. So the floor is yours. Go ahead, please.
Thank you so much. Hi, everyone. My name is Roudaina. I am a third-year PhD student in the University of Hydrobiology, as Karen said. I guess I will skip my first slide because it was supposed to be a presentation of my background. But I did the part of my studies in Algeria, and then I moved to France where I did bachelor and master. And then I went to Czech Republic to do this PhD, which is focused on the genomics and metagenomics of freshwater protists. And my talk today will be more on the metagenomics part, because it's the part that I started working on already. So I chose to start my presentation with this very familiar tree. It's actually a phylogenetic tree and the Carl Woese classification of the 3 domains of life. So all of the living organisms on Earth can be classified in one of these 3 branches. They can be either bacteria, archaea, or eukaryotes. And today I will be focusing on the eukaryotes, as you know. Even though the tree changed a little bit, it looks more like this right now. I chose to start with a very familiar tree for everyone, just to understand and put you on the same mood. So if we zoom on the eukaryotic branch, we would see something like this. So this is the tree of eukaryotes, and over here you can see the animals and the fungi, and also in this branch in Archaeplacida you can find the brown algae, and you can find the more famous organisms. But actually most of the branches belong to protists or unicellular eukaryotes. So these are single-cell organisms that can be heterotrophic, phototrophic, or mixotrophic. So they have various modes of nutrition, but they are also very diverse phylogenetically. So when we say protists, we do not mean one branch or one taxa. We just mean a multitude of taxa that are very diverse phylogenetically. And as you can see from this tree as well, most of the diversity of eukaryotes come from protists. So they are very important for studying the environment and being interested in particular environments such as lakes or oceans or wherever it is. They are the most dominant form of eukaryotes. Last year, we wanted to see what is the number of published genomes of protists on NCBI. So we downloaded all of them and we found 1,137 genomes. Most of these genomes belong to human pathogens and plant parasites. This is because of their importance to the human health and to the economy. However, these organisms are are not as dominant as the free living organisms. So we need more genomes and we need more cultures of these free living organisms. We did another type of filtration on these published genomes after downloading them. So we removed the small fragments and the very little genomes. So these are genomes that are less than 5 MB, because traditionally a genome of a eukaryote is more than 10 MB. And then we calculated something called the completeness. Now the completeness is the calculation of the number of monocopy gene markers that are shared between genomes of the same taxa. And there is also another calculation which takes under consideration the genomes of all of the eukaryotes. Now we compared these genomes using the gene markers of eukaryotes, of course, and we found that 25% of the published genomes are less than 50% complete. So even the genomes that come from isolates, that come from cultures, are not very complete. And there are, protists are surprisingly less studied. And there is another problem that comes with studying protists and trying to isolate them. That is, most of the heterotrophic flagellates, or these are protists that do not perform photosynthesis. They are unicellular and they have 1 or 2 flagella. They look similar morphologically. So if you try to isolate them just based on the morphology, you will not be able to, because they look similar to each other. And all of these pictures that you can see belong to the same organism, that is Spumella, but it belongs to 15 different species of Spumella. So it's very hard to isolate them just by looking under the microscope. There are other organisms, other protists that are very distinct morphologically, such as Fragillaria or Cryptomonas that you can see on the slide. But the vast majority of protists are actually more like this type of protist, that is unicellular, one cell and heterotrophic with a flagella. So there are many ways to obtain genomes. First of all, we have the culture-dependent method. That is, if you manage to isolate a protist, then you would grow it in a dense culture sequence, the genome. However, this is not always very easy, because we do not always have the correct media to optimize the growth of this organism. As I said, there is also a problem in isolation itself, because we do not know how to distinguish between different protists just based on the morphology. But even when we manage to isolate and grow, we do not, sometimes we cannot assemble the genome. And to encounter this problem of culture, there are other techniques such as single cell and metagenomics, which are culture independent techniques and could make our life easier if we want to sequence genomes. So I will start by presenting to you our data set. This data comes from a lab in Germany. It's the IGB lab, who worked on these 4 lakes that you see here on the slide for the last 10 years. They actually managed to extract metagenomes and sample these lakes every month for the last 10 years. And they managed to obtain more than 1,000 metagenomes for each one of these lakes, which is great work. And they were very nice to share their data with me to analyze it. So actually, the lake water was sampled from the epilemion and the hypolemion of each one of the lakes. These are the upper layer and the bottom layer of the lake. And then water was filtered through 2 filters. The first one is 5 micron. This is to separate the larger organisms such as protists. And and also the particle-attached bacteria. And the second filter is more to detect bacteria and free living organisms. After doing this, DNA was extracted, of course, and sequenced, and part of the data analysis was done in the lab in Zurich. This is the assembly and the binning of the metagenome. And I will explain to you the binning part because it's crucial to understand the results. So as all of you know, metagenomics is taking genomes from the environment and then sequencing them together. In this case, we obtain reads. These reads are assembled into contexts. And then using three main metrics, the contexts are separated into different batches. And these are the contexts that we believe come from the same organism are separated into batches, which represent one single genome. These metrics are tetranucleotides, GC content, and coverage. We can go into details later. If you do not understand, please ask me. But basically, these little batches which contain contexts that come from the same organism are called bins. Once the bins are decontaminated and we remove contamination that comes from contexts that do not belong to this bin, we call it MAG, or metagenome assembled genome. So this is where our part of the data analysis comes, and I will start explaining it to you. So we started with bins that have a size of 10 MB or more. As I said before, this is the normal size of a eukaryote, so we only started with big bins because they are considered to be eukaryotes. And then we wanted to filter them based on the context taxonomy. So we predicted the genes inside of each one of the contexts, and then we compared these genes to the database. And to do this context taxonomy, we tested 4 different databases that are commonly used for taxonomy assignment. So UniRef, UniProt, MMETSP. So this is a marine microbial eukaryote transcriptome database only. It was a project done in 2014 based on Tara. And it produces a huge amount of transcriptomes for Protists. So this is a really good database. And then we also used GTB. So we realized very quickly that UniProt and this microbial transcriptome database are very, very good. Actually, MMETSP retrieves more eukaryotic contexts than any other database. So it retrieved, in this case, for example, 222 MB in comparison to less than 1 MB from UniProt. However, different databases have different organisms, so sometimes it's better if you use more than one database. And that is what we did. We decided to merge UniProt and MMETSP using a program called MMSeq. And then we performed our gene-based context taxonomy again. This is assigning the taxonomy to each one of the protein predicted in the context. And then we started our decontamination. So first of all, we removed the contexts that come from viruses and bacteria because we are not really interested in them. And then inside each one of the bins, these are only eukaryotic bins. We found that sometimes they are contaminated with contexts that come from more than one eukaryote. This is because the metrics that I told you about before, the GC tetranucleotide and the abundance are not perfect for binning eukaryotes. So this is why this extra step of decontamination is used. And for doing this context removal, we only used bash scripts, so scripting. After doing this, we had very high quality, very clean mags. And then we calculated their completeness, and we got rid of the little genomes, and we started our taxonomy to understand which kind of organisms we have. This is an example of a bin that is contaminated with contigs that come from more than one eukaryote. So as you can see here, the bin name is 26, and these are the different contexts. And so, as I said before, we found the protein and then we assigned the taxonomy. So most of the, let me just find a little laser. Okay, so most of the contexts come from chlorella. So this is a bin of a chlorella. However, sometimes we found that there are some contexts that come from this uncultured eukaryotes. And in this case, this is considered as a contamination. So, some numbers. We started with 1,176 bins that are more than 10 MB. We removed the little contexts that do not add a lot to this study, because they are little contexts that disturb the process of filtration. We ended up with 719 bags that are only eukaryotic, and then after doing the filtration based on the completeness and based on the size, we have 280 mags. These mags belong mainly to very dominant organisms in freshwater lakes. So as you can see here, we retrieved genomes that belong to Daphnia, to ciliates, to ochrophytes, chlorophytes, haptophytes, and even fungi sometimes. And then after doing this genome retrieval, we wanted to increase the completeness. So we calculated the average nucleotide identity. This is the identity between two different genomes that come from the same taxa, to see if they belong to the same species or not. So if they come from the same species, then it's the same genome, and we can co-assemble them together. If we co-assemble two genomes together, then we increase the completeness and we increase the quality of the genome. So as you can see, at first we retrieved two bins that have different size. One of them is 14 MB and the second one is 22 MB. And then we only co-assembled the genes that are found in one genome and not in the other one. This is how co-assembly works. And we ended up with a co-assembly that is 19 MB and 78% complete. If you when we did the taxonomy, we saw that this genome is closest to Chlorella desiccata. So when we checked the completeness and the size of the genome of this species that is found from isolate, that is extracted from an isolate, we found that it is 21 MB and it is 87% complete. So as you can see, the genomes that are extracted from metagenomes can be sometimes as complete and as good quality as isolates. However, isolation and genome sequencing from culture stays the best technique for genome sequencing. I just want to say that. So when we added this second step, we ended up with 78 mags that belong to 8 different taxonomic groups, and we started performing the taxonomic assignments in order to see which published genome or transcriptome groups or clusters with our extracted mag. So in this case, we collected all of the transcriptomes and all of the published genomes. In NCBI, we verified them for contamination by bacteria or by other eukaryotes using our technique that I showed you before. And then we predicted the genes, and we made a phylogenetic tree using concatenated genes to see which one is closely related to our mag. So this is an example of a Euglenozoa mag that we have retrieved. It is 50% complete, and it is closest to Bodosantans. So that is published over here. Another example is this Haptophyta mags that we retrieved. So we found that they are 30% complete, and they cluster with Chrysochromulina todini. This is a very known freshwater haptophyte. So this result is not surprising at all. So I think I will finish this presentation way more than planned because I spoke very fast. Sorry, but we reached the conclusion right now. So from this study, we have customized a very good database for retrieving eukaryotic contigs from metagenomes. We also retrieved 79 non-redundant, high-quality Protist genomes from metagenomes. So this is the largest scale study done on freshwaters using metagenomes. There is two other or three other papers that are on metagenomes, and most of them are on marine environments. The famous Tara study. So yeah, this is the largest study done on freshwater metagenomes. Also, we noticed that some of the mags that we have retrieved are as complete and as good quality as genomes that are extracted from isolates. And I have to add also that metagenomes is a very good technique to retrieve genomes of very abundant organisms. The problem with this technique is that we cannot assemble the genomes of less abundant organisms because, as you saw, binning, during binning, the abundance is a very important metric. And yeah, thank you for your attention. Do you have any questions? And sorry, I spoke very fast.
Thank you very much, Roudaina. I don't think you were very fast, don't worry. It was all very clear. And it seems that you've done a very amazing study here. I'm totally not from this topic, but I see that people are very impressed here. Yeah, and maybe there are some questions. Please go ahead.
I will just open the chat because, yeah, okay. Thank you, people. Yeah, do you have questions? Did you understand the, yeah?
Yeah, please go ahead. I also cannot really see, maybe not if, go ahead if someone has a question. But if this is not the case, then I think your presentation went just too well.
No, thank you for the opportunity. I really enjoyed this. Thanks a lot.
Yeah, so I don't really have a clue of that, but my question would be, so I guess you are, it seems that you succeeded in what you wanted to do, and you could complete these the genomic data that you need to understand these organisms better. You were able to do that, right?
Yes. So the protists are abundant eukaryotes, but the problem is they are not as abundant as bacteria. So when we try to sequence the whole metagenomes, we obtain a lot of breeds that belong to bacteria. Yeah. And this is what makes us only retrieve genomes of abundant organisms. But if the filtration and the sampling is, the filtration of the metagenome and extraction of the DNA is done differently, in a way that we can lower the number of bacteria that are retrieved, we can maybe even detect less abundant organisms. So yeah, what I'm trying to say here is that when we do not have, when we cannot really isolate protists, then maybe the solution to this problem is metagenomes. And I think our pipeline is good for that.
That sounds very good. Wish you good luck with that. And with that, I will really thank you very much for the presentation here.
Thanks a lot. Yeah. Thank you, Mina, for telling me about this seminar because I'm very happy to present here. Yeah.
Awesome.
Okay.
And now I would like to introduce you to our second speaker of the day, who is Anwesha Ghosh from India. Anwesha is one of the very engaged members of the ASLO committee, ASLO Early Career Committee. She's basically part of our team here. She stepped in since we had a little short-term program, swap and change. And Anwesha is also working on very tiny organisms. She did a PhD in biological sciences at the Indian Institute of Science and Education and Research in Kolkata in India. A postdoc at the Center of Climate and Environmental Studies, also at the IISER in India, and now is in Plymouth, UK at the Marine Laboratory there as a fellow in a joint program of the Partnership for Observation of the Global Ocean and the Scientific Committee of the Oceanographic Research. And her work focuses on complex microbial communities, and especially with a context of ocean acidification. Anwesha, the floor is yours.
Thanks, Karen. Can you hear me all right? Okay. So, and can you see the screen?
It's not in presentation mode yet.
Yeah, go to presentation mode. Is it better now?
No, yeah. Great.
All right. Perfect. Thank you for moderating the session again, Karen. It's always good to speak to everybody. And today, what I'm going to be discussing is a global problem that we call the evil twin of global warming. And I'll try to explain why we call it so. So this is about ocean acidification, but today the angle that I'm going to show you is going to be slightly different from that of the open ocean and the acidifying problem that we know today. And today my focus is going to be on coastal oceans. This is a work that we've been doing in collaboration between the UK and back at home in India. So to start off, did the screen change at your end?
Yes, it did.
Yeah, perfect. So what you are seeing here is what we know ocean acidification as. We add more and more carbon dioxide into the atmosphere that dissolves into our oceans and essentially produces more H+ ions. This is the problem as we know it today. What has been the issue is that the level of carbon dioxide in the atmosphere has been rapidly increasing. Consequently, our oceans are taking up more and more of the atmospheric CO2. This would not be a problem, and to be honest, this was not so much of a problem till now, but unfortunately now our oceans are reaching maximum capacity for carbon dioxide uptake. Consequently, what is happening is that you will see in the panel here that when more and more carbon dioxide starts to dissolve, and this is a problem that we are seeing for the last decade, that we not only see the dissolution of the calcifiers, what we also see is that these calcifiers are becoming smaller and smaller in size. So what is the problem really? We don't mind having a lot of H+ into the oceans. It is probably something that the organisms can combat. But what is happening is that this there are specific forms of carbonate that the oceanic pH is actually altering. And one of the primary factors that it alters is the calcium carbonate component. And what we measure it is, we measure it as calcium carbonate, and we also measured it as aragonite. And as you can see here is that the aragonite concentration is very rapidly increasing. Now, aragonite is the most soluble form of calcium carbonate, and this is the form that most of the marine calcifiers use. So here what you see is a coccolithophore, we have the mollusks, we have the gastropods, and these shells are all made of calcium carbonate. Now, the decrease in pH is essentially starting to dissolve these shells. We do not see a change in size and shape, but what we do see is that the cells are becoming more fragile. So if you hit the shells harder, they are probably going to end up breaking. So why did I start off by saying that this issue of ocean acidification is slightly different in the coastal oceans compared to that of the open oceans? What has been happening is that the coastal oceans are already very used to rapid changes in pH, because the coastal ocean has two main forces that balance the pH apart from the carbon dioxide that is being dissolved. Most of our coastal oceans actually do have very high freshwater input from the land. These could be coming from rivers when the coastal oceans span over estuaries, or they could be coming from lakes or adjacent terrestrial runoff. But what also happens is that the oceanic pH in the coastal ocean is being balanced by the tides bringing in marine water, which is essentially what causes the buffering action. Most of the fragile ecosystems also are located in the coastal oceans. As you can see here, many of our coral reefs are very close to the shore. So consequently, the way we see global ocean acidification on the coast is slightly different than we see in the open ocean. So we were doing part of the research where we were trying to see how the mangrove ecosystems essentially behave when we put them under the global ocean acidification problem. And this is an ecosystem known as the Sundarbans, where I did my PhD. So it's located in the eastern part of India. It spans across India and Bangladesh. This ecosystem is about 9,400 square kilometers of area, very rapidly reducing in forest cover size, partly because the entire tectonic plate is starting to subside. Now, this region sits over the Ganga-Brahmaputra-Meghna Delta, which is the second highest freshwater source into the coastal oceans. And what you'll see here, all of this brown essentially shows you that there's a lot of sediment in the water, making it very difficult to study. This also sits on the tropic, as a result of which you have very hot summers, not very cold winters, but we have a very distinct seasonal pattern and a very distinct wet season, which is the monsoons. So what happens because of the monsoons and because of the diurnal tides in this region is that you have a lot of water movement happening at the same time. So there's a lot of mixing that happens. So at any point of time, the dynamicity within this region is extremely high. But at the same time, even with environmental conditions being very difficult, this forest is home to one of the highest biodiversity regions. What you will see here is we have a very distinct gangetic dolphin that survives in this region. You have the Royal Bengal tiger, which is also India's national animal. Now, all very great, but this ecosystem has been facing a very rapid challenge in the sense that the ecosystem is fast degrading. It sits on a hotbed of tectonic movement. And when we go on to do research, it is extremely difficult to conduct research in this area, partly because you are in a tiger habitat. So you have a chance of being attacked by the tigers. And unfortunately, the tiger is a national priority. So your research does become secondary. But this region is also infiltrated by the saltwater crocodiles, which is also endemic to this area. And every time that you're in the boat, and if you're in a small boat, there is a chance that you'll see a crocodile just swimming by. So as I said, it was very important to monitor this region, and there has been regular samplings that we've been doing. We do have a time series right here based on this estuary because it has the highest freshwater flow. And we do a monthly sampling where we measure a bunch of parameters across the sampling time frame, and you can see all of these listed here. So when we track the pH of this region, and we have 3 stations, as I've shown you here, there's another station over here, which is marked as station 1 to 3. So you have river water coming in from here, and the saline water from the Bay of Bengal comes up this region and into this creek. So you see that during no tidal conditions, so during low tide, you have absolutely no water in the creek. But then when the high tide comes in, the water level increases by 5 to 7 meters, and this is happening every 6 hours. So when we try to detrend, so we essentially remove the seasonal pattern from the pH data that we had. What we see is there is in fact a steady decrease in pH. This is still not significant, but this might become significant in the coming years. So we did a very simple experiment, which was a little different than the general pH experiments that we do. So what we generally do for pH measurements is we hypothesize that the pH has changed, and the pH is going to remain so throughout the experimentation period. But that is not what we did here. What we did is we did the experiment for a short span of time, because the amount of dynamicity in this region essentially means that your conditions are changing too rapidly. So we did not want to stretch this experiment for very long, and we only did it for 21 days. But what we did is we only altered the pH in the beginning of the experiment. We increased the pH to a certain level because it still falls under the pH range of this region, and we decreased the pH in the other set of experiments. So what you will see here is, let me show you this first. In the control containers that we had, in the control mesocosms, you will see that pH does not vary significantly, and the range is still very short. Whereas when we increase the pH, the data points were in multiple places and the data variability was much higher. When we decrease the pH, the data variability was lower. But when we did this, look at the orange curve, which is increase in pH. The pH did gradually come back down. When we decrease the pH did again go back to its normal levels, but it took much longer for that to happen. So if I remove the control, this is what it looks like. My increase in pH also comes down to my control conditions. My decrease in pH also does come back to my control conditions. And we predict that this is happening because of the biological community's capacity to alter pH in ecosystems. Though this seems very small scale in large scale ecosystem, because your ecosystem level processes are much higher. So your change in temperature or a tidal condition is going to alter pH much faster than the community does. But nonetheless, the community is capable of also altering the pH. So this ecosystem is essentially diatom driven. We have very few dinoflagellates. So what you will see here under the control conditions, high amount of diatoms, lesser amount of dinoflagellates. Conditions do not change with an increase in pH. Your diatoms still dominate, but you do have the dinoflagellates coming in. But with the decrease in pH, you see that the diatoms go down and the dinoflagellates keep coming up. So we predict what could possibly be happening is that in the near future, the ecosystem can go from being diatom driven to being dinoflagellate driven. And you don't need to focus on this graph, but it essentially does tell us that the diversity is going to go down with a lowering in oceanic pH. And even if you do not focus on the individual groups, you can simply look at the legend and you'll see that the legend is much shorter here, which implies that the diversity is lesser when the oceanic pH is lower. So coming back, we wanted to see if what is essentially being affected by this pH change. So what we saw is among a lot of other nutrients, nitrate had maximum amount of variation because of lowering pH. And you will see with altered pH condition, the nitrate level keeps going down. Now, this can have a multifaceted explanation in the ecosystem with the change in the community structure. Primarily, we know that nitrate is a preferred nitrogen form by diatom. So with the decrease in nitrate, it could be that we have a higher functionality, even with lower diversity. So this topic is still very up for debate. But what we did see here is, so when I plotted the decrease in pH versus the increase in pH, the green one showing you the increase in pH, you will see that you have a distinct community difference. There is a possibility that we would end up with a more specialized community structure if pH keeps on decreasing in the same way in business as usual conditions. So what we did next was we wanted to see if productivity was in fact being affected, because it's okay that the community structure changes, but we also know that there are groups of diatoms or dinoflagellates that are functional relatives. So we know that if one form of diatoms vanish, it most likely might be taken over by some other type. But we have very little information to actually go ahead and prove that. But what you will see is that if we take the control to be normal conditions, any changes in pH is going to affect productivity. Be it an increase in pH or be it a decrease in pH, it is going to lead to ecosystems being less and less productive at a microbial scale. Consequently, you will have an effect on energy transport, nutrient transformation, and consequently your fish yield is going to go down. So our best hope is for ecosystems to remain in this condition. So what we did is we wanted to see if this clustering pattern was being reflected, and we did a very simple NMDS test, and you'll see that all of these groups here is the start of the experiment. And as we move more and more away from the start of the experiment, it essentially starts to cluster together. And it was difficult to differentiate this in this NMDS scale. So I simply just teased this out, and you will see that this bit of the scale has been expanded here. And you will see that more of the days start to cluster together, again indicating that the community does appear to go back to its normal condition, probably bringing in community level adaptations. Again, I see this with caution because we don't know the scale at which this adaptation can start to come in. But given that coastal communities are very used to this kind of dynamic situation happening, they may be far better equipped to handle ocean acidification compared to other groups. So we went ahead and did some GNM modeling, and it also gave us the same kind of information that if we do continue these stress conditions, we will see that the organisms do still come back to a normal condition, which is what we see under control conditions. So even if I alter my pH conditions, I would see that organism level adaptations might actually help the organisms come back to its own form. So briefly to conclude, so starting with the photos first, you will see that these are some of the diatoms that we see. What you see in the background is because of the high suspended particulate matter, which makes it very difficult to do any level of microscopy here. We also do molecular data, but then as you heard in the previous talk, the caveat with the molecular data for eukaryotes is that the databases are not as enriched as that of the bacterial data sets. So you would end up getting a lot of hits which tell you that the database cannot identify what you're seeing. But using microscopy, you can still identify, you can get an idea of the shape, cell shape, size, etc. So the best approach would be to actually use microscopy and molecular techniques together. But to briefly conclude, what we saw is that all diatoms do not behave alike under ocean acidification situations. Obviously, given the generation time of diatoms, the shape of the diatoms, the genome size, makes it more resistant to changes or more susceptible to changes, and which is where the question of adaptability really comes in. What we saw is that the coastal phytoplankton might be capable of regulating coastal pH changes, but then again, this is difficult because these organisms are very small and the ecosystem as a whole is very large. What we saw is that the dominant members might be able to survive such changes, but the rare taxa might be lost out. So that's where your community becomes more specialized type. So we lose out on the general members, and we don't know what kind of an effect a specialized community will have on downstream factors of coastal nutrient transformation, etc. What we don't know is what happens to factors for nutrient availability. We don't know if a decrease in pH is going to affect productivity in large scale, or if some organisms will, have a quicker kickstart to productivity and they can increase or boost the productivity. We obviously need to link this to nutrient availability and uptake rates, which is something which is very difficult to measure, and hence we usually bypass doing these measurements in large-scale experiments. And lastly, what we saw is that the dinoflagellates tend to substantially increase in number under low pH conditions. We don't know if this is going to be happening in the ecosystem overall, but if we take time scales of hundreds and thousands of years, it is a possibility that you'll have a higher dinoflagellate number in coastal ecosystems. Thank you. So there was a lot of money which went into this grant, and this is work that I presented in Spices. So yes, that's the end of it. Thank you very much.
Thank you, Anwesha, for this again very, very in-depth insights into this ecosystem also that we've heard a lot already in this seminar and this webinar, and it's again very impressive to see how much work is done and what work you do there. Very amazing. Are there any questions about that?
Yeah, I think Alba has a question.
Oh, there's one question, yes.
A question
did you measure dissolved organic matter concentrations? And if yes, did it change with different treatments?
Yes, it did. It did. We did measure TOC. We actually measured TC and TOC and then the dissolved fraction using a carbon analyzer. And what we have seen is that there is a decrease in DOC, but remember in coastal oceans, what happens is that there is a higher amount of allothonious carbon compared to autothonious carbon in the ecosystem. So if you try to draw parallels between what is actually happening in the ecosystem versus in your mesocosm setups, it would be difficult to say. But with the DOC measurements that we had, we saw a decline in DOC.
Okay, thank you. Are there more questions? And also, please go ahead and just jump in and ask. If that is not the case, I would just quickly, out of my just interest, also ask, like you said that you saw that the organisms are kind of able to reverse the situation, and I wonder, in the long term, would you think that you would also have a change of organism and of community change, and that you would have communities that are better able to cope with the more acidified conditions?
Yes, unfortunately, we don't know. We can't say. Because a lot of these experiments, I mean, under situations where we have done long-term mesocosm experiments, we have seen the community crash. But that could be happening because of XYZ number of factors. We can never do it for a time scale long enough to capture adaptation, right? But if you think about it in the ecosystem, when your daily changes in pH ranges by a 0.5 plus minus units, it's obvious that the organisms are already used to this change. They have that adaptation. Their genome probably encodes for proteins that can survive in this large pH change. For example, in the last 50 years, what we saw is that diatoms have now two different kinds of nitrate transporters. They have a high nitrate transporter, they have a low nitrate transporter. We don't know if these nitrate transporters were already existing in the genome, or the organisms have now started to just express those genes in order to combat stress.
Yeah.
So we have very limited studies which actually throw light on organism level adaptations. The max we can say is that the community structure is changing, and unfortunately that's where our information ends. So I guess when we move our focus from the large effect of ocean acidification to actually go down to more fine scale experiments, can we probably have some predictions in hand? Yes.
No. Great that there's so much work done on that, and thank you also to share your results with us, both of you, Roudaina and Anwesha. Thanks a lot for this last Amplifying Voices webinar today. I don't see more questions, I think, for Anwesha, or also maybe still for Roudaina. Then I would thank also the audience for joining us here today. And we will be in touch with you as soon as possible to make sure that you all get all information needed to join us in September again. Thanks a lot and bye-bye.